The most important part of this is you guys participate and interact. When I ask you questions, answer them, give me your thoughts, etc. That will be helpful. If you're all just gonna sit there and look at me like, what happened, man? This isn't gonna help. Okay, we'll just do work. But if you guys wanna participate, we'll talk about stuff, I'll spend all day doing this. All right, fair enough? Question 1 is the same on both forms, but the answer choices are different. Not different answers, like A is C and B is D and stuff like that. You all understand what I'm trying to say? So it's the same answers, they're just different letters. That prevents you from looking at the nearest Scantron and just Christmas tree that in. Like the nearest Scantron. Every test up here is fun. So 30 of you took the test. I always do an item analysis to find out what questions you guys did well on, what questions you guys did not do well on. I refine these tests. That's why you can't take them with you. You can always come and see them in my office, but you can't take them with you. We're going to go over them today. If you want to see them again, you're more than welcome to stop by my office. But I assure you that none of these questions are the final. So I'm just going to study the test because it's going to be the final. No, that's not how it works. All right. So question number 1. I like to start off a test with a question that I think is going to be pretty easy. 18 of you missed this one. That's more than half. I want you guys to all open up your textbooks, page 4. Highest level you expect a blood vessel to be. Are blood vessels organs? Yeah. Yes. Remember I showed you that picture and said, look, oh my God, a blood vessel is an organ. And I used the example another time too. If you got that wrong, how many of you picked organ system if you got it wrong? All right. How many of you picked tissue? Okay, that's good. Looks like most of you picked organ system. Pretty sure nobody picked cellular, right? If you did, don't put your hand up. Organ system or organ, those are your two choices. What would it have to be to make organ system the right answer?
2 or more organs.
2 or more organs, yeah. There's a bunch of blood vessels. And the heart, yeah, that's an organ system. If it's just a bunch of blood vessels, well, those are all organs. Does that make sense to you guys? Do you understand now why organ is a good answer?
I just didn't read the question. I just really didn't.
You're gonna find that happens frequently, and there's a couple more in here that may have gotten you. And I'm sure there's a couple in here that nailed a lot of you. One of the things I do with my tests is try to get you guys to develop a degree of precision. You have to read all of the words. I'll say this again, but most of you guys are going into healthcare careers. If you make a mistake by not reading what's on the piece of paper in front of you or the screen or whatever, you can damage people, you can hurt people, and in bad cases, you can kill them. Medical errors. Happens all the time. I don't want you guys to have to go through that. I know people who've made med errors and it impacted patients, and that sucks. So you're gonna be, oh man, yeah, about the test questions, but I'm doing it for a reason, okay? So, that was number 1. Number 2, 7 of you missed number 2. You got your home air conditioner in terms of homeostasis, the wall thermostat. What's that gonna be? That's the control center. Remember I drew you that little picture up there on the board? A lot of times when I draw you pictures and stuff, and when I say something over and over, that's a clue to you guys. Hey, pay attention to this thing. You got a question? Yes.
Well, I would have gotten it right, but I second guessed on the question, so.
Yeah, that happens a lot. I want you guys to kind of go through and think to yourself, ooh, I changed this answer from a right answer to a wrong answer. Watch for that, because I bet a lot of you have done it. If you went through tests, you come up good. If you went through tests, you come up good. Then that's that. A lot of you guys, how many already know that you changed answers from right to wrong? Yeah, stop doing that. I know it's easy to say, but I normally talk about this a little later in the gig, but hey, since we're here now, well, I started taking multiple choice tests, a lot of them when I went to Penn State. Like my senior year classes had 300 people in them. My undergrad classes, I had an undergrad bio class where there were 500 people in it. It's like, holy crap, this big auditorium. Everything was multiple choice. I could probably justify every answer on that paper if I thought about it long enough. So I would lose points. Well, maybe he means this, maybe she means that. The way I got around that was I tried to take the test as fast as possible. First impression, boom, done, never look at it again. Don't go back, unless you're getting information from other questions. I got pretty good at it. I can take a multiple choice test that I know nothing about and get a 60 or 65%. So if I know 30% of the material, I got an A. I want you guys to get better at taking multiple choice tests. I'm gonna tell you some little tricks to use as we go along here. All right. So that one, just remember the drawing up on the board. Control center determines what to do. All right, number 3. Wow, some of you guys had problems with number 3. 14 of you got number 3 wrong. Just about half. The purpose of negative feedback is, how many of you think all of those answers look right? How many found 2 that definitely look right? Yeah, this is a pick the best answer kind of question. You're gonna get a lot of these. If you've taken a state board test, they're all like this. Pick the best answer. They all kind of look right. I want you guys to get used to that, to get good at that. It cost, last time I checked, it was $600 to take a state board for your nursing license. I know people who flunked it 3 times. How many guys have $1,800 you can throw up in the air for flunking a test? Nah. So I want you guys to get good at this. Let's take a look at this and examine it. The purpose of negative feedback is to regulate. Does that sound right?
Yes,
Sure. How many guys picked to regulate?
I didn't.
You're right. You're right. It does regulate. But is to regulate a good function in this class? I'd say regulate what, right? To regulate is kind of okay. It does regulate stuff, but let's look at some more. Let's look to control all levels of the body systems. Does negative feedback control all levels? You can cross that one out, right?
Yeah.
How many guys cross out an answer and never look at it again? Do that. On my test, you can always write on my test form. Okay? You can always write on my test form. So if you know it's wrong, cross it out and never look at it again. It doesn't do all, cross that one out. So, to keep the body sugar normal, does it do that?
No.
Yeah, it does. Negative feedback controls body sugar with insulin and glucagon and keeps it in a normal range. So that one's pretty right too. But that one's really specific, isn't it? That's only one thing in the body. Now, to prevent sudden and severe changes within the body, does that sound pretty good?
Yeah.
Which one of those is the best answer for negative feedback? Sudden and severe changes.
What got me on that one is prevent sudden and severe changes. I thought that was buffers. And
Okay.
that's what got me. I was like, oh, that's a buffer, that's definitely not right.
Yeah, that's just pH. That's just pH. So do you guys have a better idea why that's the best answer now?
Because
Does anybody have any questions? Who thinks that's a trick question? It's okay, you can. It's fine. There will be trick questions.
so
As we
it
continue
should
on
always,
all the tests.
should always be the most specific answer.
I want the best answer. I always want the best answer. Alright, so number 4. 4 of you missed number 4. I'm not sure why. Labor and blood clotting, those are the two big positive feedback loops. If you miss number 4, I don't want to be mean to you, but kind of not getting, you're not studying the notes enough. That was flat out in the notes, okay? If you're missing number 4 there, either with an accident, okay, I gotta get it. But if you actually didn't know that, you need more study time. Number 5, one person got that wrong. I'm gonna assume that was just an error, and you missed eat something. It says homeostasis is a relatively stable internal environment within limits, period. Number 6. 6 of you got number 6 wrong. The lungs are located in the following cavities. Now, if you look through there, see it says mediastinum. If you see mediastinum for me, what should you do right away? Cross it out. Remember I told you guys mediastinum is not a cavity. So we have pleural, dorsal, and abdominal. Where's your dorsal cavity? Back here. Is that anywhere near your lungs? Is your abdominal and your dorsal in the same cavity? Does that make any sense at all? Cross it out, right? So we have left. If you can get it down to 2, you got a 50-50 shot. Straight gas, 50-50 shot. Pleural, ventral, thoracic, pericardial, ventral, and thoracic. When you see cardia, what do you think of right away? Heart. Cardio. Heart. Cross that one off. You're left with the right answer. How many guys do that? Cross all the wrong answers off and look what's left. That is a definite good tool. If you're stone guessing, you got a 25% chance. You'll probably score around 25% on the test if you're straight guessing. If you can narrow that down to 2, you're now at 50-50. You're much closer. So cross off as many answers as you can. Let's see, number 7. 10 of you missed number 7. Place following correct sequence from simple to complex. What's the simplest thing in that list? Atoms. Because atoms go together to make molecules, right? Atoms go together to make molecules. So, it's got to start with a 2. So how many can you cross out? 2 of them. You can get rid of 2. Because they don't start with atoms. Now, between those 2, organism, most complex thing there, they both end in 5. So we're down between 3 and 4, and 4 and 3. Which is more complex, cells or tissues? Tissues. There's your answer. You only had to know 2 things. Know it started with atoms and know that tissues are more complex than cells. You can break this stuff down and check stuff off. It's a great way to do multiple choice tests. Number 8. 7 of you missed number 8. Dorsal body cavity is the site of which of the following? Brain, lungs, liver, and intestines.
Brain.
Yeah, the answer is brain. But I want you guys to put your thumb where your lungs are. Right here, right? Actually do it on the right side. And then put your finger where your liver is. You guys know what side your liver's on? Right side? Put your finger where your intestines are. Do it right here. That far away from each other. Where's your brain? Really far away. So you didn't have to know the names of the cavities to figure that out. I got 3 close together, I got 1 far away. That is a great strategy for multiple choice tests. Look for 3 things that are similar and 1 thing that's different. A lot of times, the answer is a different thing. I'll go so far as to say, if you have 3 long answers and a short answer, I check the short answer. Be short answer than a long answer, I check the long answer. That's a definite drop back and punt move. If you have no idea what you're talking about, do that. Look for 3 specifics and a general, 3 generals and a specific. Look for the odd man out if you're searching for some guidance somewhere. Again, that's last ditch stuff, but yeah, that's also a good multiple choice technique. All right, number 9, functional characteristics of life is irritability. 3 of you got this wrong. Sense changes in the environment and react to them. I'm not gonna ask who got it wrong. Number 10. Everybody got number 10 right, yay, you guys, everybody applaud, you all got it right, yay. Number 11, 18 of you got it wrong. Genetic information is coded in DNA by, the correct answer is sequence of the nucleotides. Remember what I drew up here? Just out of curiosity, more than half you got this wrong. How many guys picked 3-dimensional structure as double helix?
I read the question wrong.
Okay. How many guys picked regular alteration of sugar and phosphate molecules? So how many guys picked structure of the histone? Oh, okay. Yeah, histones, remember, just like clothes hangers. They hang up the DNA so it doesn't get crumpled up. 3-dimensional structure, it is the same. The DNA structure is the same. The sugars and phosphates are exactly the same. There's no information there. The information is in the GCAT, the order that they're in, which determines which amino acid is next, which determines primary structure, which determines everything else. Number 12.
Oh my God.
15 of you got 12 wrong. Spot on half got 12 wrong. Carbohydrates are stored in the liver and muscles in the form of, okay, how many guys picked glucose? If you saw carbohydrates and said, oh, glucose is a carbohydrate, right?
I had glycogen and I said yes to myself.
Yeah. Remember, glucose is a monomer. Glucose is one circle. Glycogen is a boatload of circles, right? Bunch of glucose is all stuck together. That's how we store them. If you pick triglycerides or cholesterol, the question's asking you for carbohydrates. Are triglycerides carbohydrates? No. Cholesterol carbohydrate? No. 13. Ooh, 19 of you got 13 wrong. Carbohydrates and proteins are built up from their basic building blocks by what reactions build polymers?
Dehydration.
Monomers. You know this.
Dehydration.
Dehydration synthesis, and then hydrolysis takes it away. So you know you're working with water, right? Dehydration synthesis and hydrolysis. If you're building stuff, what is it? Dehydration?
Dehydration.
And when dehydration, do you add water or do you remove water? Remove. Remove. Does that make more sense now? 14, looks like 11 of you missed 14. Salts. This is pure memory. Always ionic compounds, ionic bonds. What makes an ionic bond? What happens in an ionic bond? One atom gives away an electron, the other one takes it, right?
Yes.
Always ionic bonds. Number 15, looks like 13 of you missed 15. Numbers listed represent first, second, third energy levels, respectively of a space which solid is an unstable or reactive. What rule are we talking about here?
288.
Rule of octets. What's the numbers in rule of octets?
288.
288. Which one of these is not 288?
See.
281, right? Now, that's how I would do it. Okay, I know rule of octets. This one's different than the rule of octets. That's probably the answer. You could approach it a different way. If an atom had 281, what atom are we talking about? Look up there. What atom? 281. What's 2 plus 8 plus 1? 11. What's number 11 up there? Sodium. What does sodium do when you put it in your pants? Catches on fire. Is that reactive? Do you remember the story I told you about little Johnny? That's another way to approach it. How many guys thought about it that way?
That's too much
Curious.
thinking.
No, weird,
Too much
okay.
thinking.
Yeah, it's got the extra electron. That extra electron is gonna wanna go somewhere. All right, 16. 8 of you missed 16. A solution that has pH of 2 would best be described as being, correct answer there is acidic. If you got this wrong, how many picked slightly acidic? At least you're in the right ballpark. If you picked basic or neutral, you should go back and look at pH again. 17, 6 of you missed 17. Long chain of simple sugars. Since we're talking about sugar, what two can you eliminate immediately? Protein and nucleic acid. Those aren't sugars. Protein and nucleic acid. So, monosaccharide or polysaccharide?
Poly.
Which one's a long chain?
Poly.
Poly. Lots of. You guys know what polyester means? A lot of esters.
Ah.
Really? That's when we first started doing polymers, long chains. Poly's many. 18, 9 of you missed 18. Coiling a protein backbone into an alpha helix. When you hear alpha helix, what do you think? Secondary structure. Alpha helix, beta pleated sheets, or beta sheets. Secondary structures. Primary structure is what? Ordered amino acids, right? What's a quaternary structure? 2 or more together, 2 or more protein chains together. All right. Now this one, I kind of sprung on you a little bit. But you did pretty well. Only 6 of you missed this one. 4 elements that make up human beings.
John.
Chon, C-H-O-N, yeah. Carbon, hydrogen, oxygen, and nitrogen. I'm from Pennsylvania, Dutch country. Mechanicsburg, PA is pretty close to Lancaster. You guys heard of Lancaster? You guys watch the, what is it, Amish Mafia or whatever the reality TV show was? I drive past some of those places pretty frequently when I'm up there. Chon is Pennsylvania Dutch for John. Hey, Chon, throw the horse over the fence some hay. I mean, that's a legitimate session structure there. So that's how I remember that. Maybe you guys will too, C-H-O-N. All right, 20, selective statement is most correct regarding chemical bonds. How do we do on 20? Looks like 13 of you missed 20. Let's do a process of elimination. Do ionic bonds share? No, cross it out. Do covalent bonds take? Transfer? No, cross it out. Are multiple bonds possible with covalent bond? Yeah. Sure, look at how many things carbon sticks to. So the one's left, hydrogen bonds often occur between different molecules and involve water. I mean, you can kind of get that from the name hydrogen bond. You guys think this is helping?
Yeah.
Okay. '21. We did pretty good at 21. 5 people missed 21. Amino acids joining together to make a peptide is a good example of a synthesis reaction. You're making stuff. You're synthesizing stuff. 22. Let's see, 12 of you missed 22. RNA responsible for bringing amino acids to the factory site for protein formation is the, who brings the goods? Transfer RNA. What does rRNA do? That's a ribosome. What's it function as? It's the machine that puts it together, right? And mRNA is the recipe, yeah, the mirror copy, the message. Just out of curiosity, how many of you guys picked ssRNA because you hadn't seen it before? Sometimes people do that. How many guys have ever picked an answer because you didn't see it and it wasn't?
I have.
Yeah, don't do that. Trust yourselves. ssRNA is a thing, and single-stranded RNA, there's a category of viruses who use single-stranded RNA to pass their message on to the target cells. So it is a thing. It's nothing we've talked about in class. If you come across something on a test that you've never seen before, cross it out. I mean, if they get you, they get you. If you made a mistake, own it. But a lot of times people will throw weird stuff in and hoping to catch you. Just cross it out if you've never seen it before. Trust yourself. You guys have all spent enough time studying this stuff. Speaking of time spent studying, how many guys are going to study more for the next test? All right. All right, 23 usually gets a fair amount of people. In this case, we got 17, right around half. Red blood cell placed in pure water. How much dissolved stuff does pure water have? None. So is that hypotonic or hypertonic to a red blood cell? Hypotonic. What happens to a red blood cell in a hypotonic environment? Right? How many guys got messed up because you thought pure water had stuff dissolved in it? It's okay if you did. Yeah.
So I said, I set that one up.
Yeah. $24. Let's see, 8 people got 24 wrong. I'm sorry, 9 people got 24 wrong. Oh, by the way, I put an order in to get a new ink pad on the Scantron reader. I know your Scantrons are pretty light, the little pink stuff. Y'all see where your grade is, right? Put that number over 50, that's your grade. The letters are the right answers beside each one. All right, 24. Which of these is not a function of the plasma membrane? How many messed up because it says not and you forgot it said not? That's another thing that happens. You got to pay attention to that. When I have a not question, what I'll do is cross out the not and then cross out everything that is. If I find something that is, I cross it out. So I'm left with the not. That's also a tactic that I use. By the way, how many guys have ever heard it's never none of the above, always all of the above? Again, drop back and punt. Like, don't pick that first. But if you're confused and you're not sure, never none of the above, always all of the above. All right. Now, I know that trick, so don't use that on me. But, all right, so function of plasma membrane. Is it selectively permeable? Yeah, cross it out. Because we already crossed out the not, right? If you guys want to do that, you can do that on your test right now. Cross out the cross out selectively permeable. Does it separate the cell from the outside environment? Yeah, cross it out. Does it act as a site for cell-to-cell interaction and recognition? Yeah, cross it out. You're left with 1. Does it prevent potassium ions from leaking out of the cell? Oh, that's the answer. How do we know it doesn't prevent them from leaking? Because there's a sodium potassium pump that put them back where they belong. Right? $25. 8 of you missed 25. Cell junction that promotes coordinated activity of cells by physically bonding together in the cell community includes all the following except How many guys got messed up by accept? That's another thing that happens. You forget the accept. Oh, look, gap junctions is first. I got that one. So we got gap junctions, desmosomes, tight junctions, and peroxisomes. What's the oddball there? Peroxisomes. All right, 26. 13 of you missed 26. Which of the following is a function of a plasma membrane protein? Let's cross some stuff off. Are plasma membrane proteins circulating antibodies? No. Circulating means flowing through the circulatory system, right? And we're talking about a plasma membrane protein. It can't be that. Do they transport oxygen? Oxygen diffuses. The plasma membrane proteins form a lipid bilayer. No, they're not lipids. Proteins. How many guys picked that because it said plasma membrane? Yeah, it's okay. I mean, that's, you see plasma membrane goes with that. Yep, I totally get it. But you gotta slow down a little bit, read the question, make sure you're reading the whole question. Molecular transport through the membrane. Now, how do we do on 27? Ooh, you guys did well on 27. Only 70 missed 27. Sometimes this is one that messes people up because they say mitosis and they all say formation of two cells. If you got it wrong, how many guys picked formation of two cells? Yeah, remember a couple times I said it's the nucleus, it's division of the nucleus, nuclear division, blah, 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 blah. Yeah, cytokinesis is making two cells, mitosis is making two nuclei. When I harp on stuff like that, put a little star in your notes. Because if I'm making points and talking about it for more than 5 seconds, it's probably important. Looks like 11 of you missed 28. Passive membrane transport. Let's see. From an area of low to high concentration, is that passive? No, cross those two out. Consumption of ATP, do I need ATP for passive processes? No, cross it out. You're left with movement of substance that was concentration gradient. Passive process, no energy, downhill. 29, looks like 10 of you missed 29. Are mitochondria always the same shape? No. Is anything in human beings always the same shape? No. Look around the room. Do mitochondria make peroxisomes? Nope. Do they make proteins? Nope. Only thing left, they used to be bacteria and carry their own DNA, and I talked about that for a little while too. You remember me talking about that? Again, if I babble about something, usually you're gonna need it. Number 30, 14 of you missed 30, so just about half. DNA replication. Is DNA replication mitosis? How many guys picked that? Yeah, do you know why it's not the case? DNA replication actually happens before mitosis. Mitosis is making two nuclei. Remember the S phase of interphase? That's where DNA replication takes place. So it's during interphase, during the S phase of interphase. All right, 31. 18 of you missed 31. So a little more than half. Which is following as a principle of fluid mosaic model of cell membrane structure. The lipid bilayer is solid at a body temperature, thus protecting the cell. Are we solid? Are we like trees?
No.
No. Trees have cell walls. We don't. We're squishy. So if you got that wrong, how many of you picked Phospholipids consist of a non-polar head and polar tail with 3 fatty acid chains. Put your hands up, own it. Why is that wrong? Because there's only 2 tails, not 3. Some of you guys think that was a trick question. It's okay. So if you read the doctor's order, it says give them 3 pills and you only give them 2. Is that gonna be a good thing? No. Read the whole question. There's gonna be another one you guys are gonna go, in just a minute here. But that's not right. The phospholipids form bilayer, largely impermeable water solid molecule. Number 32. 60 we missed 32. Functions of centrioles include, do they make ATP? No, cross it out. Do they serve as a site for ribosomal RNA synthesis? Cross it out. Do they move stuff on the cell surface? Cross it out. My time is spindle. 33, 17 of you missed 33. Crenation is what happens when you put the blood cell in a hypertonic solution. It goes like that, and shrinks. That giant sucking sound. I did that for you guys, right? When I do stupid stuff like that, I want you to pay attention. Go ahead.
I put B, but it marked it wrong.
Okay, did you maybe have 2 things on the Scantron?
Yeah, I
Alright, when I'm done, come and show it to me.
Okay.
But understand that you're not gonna get to see it on the final.
Okay,
So if you keep erasing stuff, you might lose points on the final.
okay.
I mean, I'll fix it, I get it. How many guys made an error moving stuff from the paper to the Scantron? Like a wrong number or something. How many guys erased the Scantron answer?
Yeah.
And, okay, let me show you guys how I take tests. I go out number 1, and there's the question, and then A, B, C, D, blah, blah. What I'll do, when I first get the test, I'll start doing this to start like normal, and I won't spend more than 30 seconds on a question. If I look at number 1, I got no freaking idea what that is. I go to the next question. How many guys sit there and will churn on it for like 2 or 3 minutes? Oh, what is this? What is this? Don't do that. Skip, go to the next one. I have no idea what one is. I go to 2. Number 2, I know I can cross out those 2, but it might be C and D. Okay, cool. I'll come back to that. Number 3, oh, it's definitely B. And then I'll write B over there, circle B and put it there. And all this time I haven't touched my Scantron. I'll go through the whole test that way the first time. Answer the ones I know, don't answer the ones I don't know. That does a couple things for me. It lets me know how long the test is. And also lets me look through the whole test. How many guys have picked up information about one question from another question? I call that data mining.
Yeah.
This way, you go through the whole test and you know what's there. You can kind of compare questions and say, this kind of helps me out with this. So if you go through the whole test that way, good. So coming back again, still don't know what number 1 is. But number 2, I picked up some information, so I'm definitely going to C here. So I'll put a C there. And go through it again. Here's number 1 again. Okay, I gotta take an answer. Well, A is really long, B, C, and short, so I'll pick A. I have no idea what it is, but we'll pick it because it's the odd man out. Way there. And then, when I have everything done on my test sheet, then I'll start doing the Scantron. Like that. That way I don't erase anything, I don't miss stuff. I've had people where they coded an answer twice and then the rest of the test was wrong, stuff like that. That eliminates all that. You don't have to do it that way. It helps me. It may help you, all right? So, 34. Looks like 15 of you missed 34, so just about half. Which of the following is not found in the matrix of cartilage, but is in bone? Blood vessels. Bone is vascular. Cartilage is avascular. And they both have lacunae, they both have organic fibers, they both have live cells. But they don't have blood vessels or cartilage. That's why it takes so long for cartilage to heal, remember that? All right, 35. Looks like 13 of you missed 35. The blast cell for blood cell production. Well, not really sure we talk so much about those guys, but what's a fibroblast make? Fibers. What's an osteoblast make? Bone. What's chondroblast make? Cartilage. What's left? Hemocytoblast. How many guys knew that hemoglobin is in blood? How many guys picked it because of that? Good job. 36. 6 of you missed 36. Epithelial tissue. How's the basement membrane? You can get rid of acellular, right? You can get rid of vascular, right? So, contains a number of neuron types. What the heck is that? That's a wacky answer, isn't it? Do we talk about that? Ignore it. Basement membrane. 37 got some of you guys. 21 got 37 wrong. Which is following will be the most importance in goblet cells and other glandular epithelium. What do goblet cells do? Make mucus. If you're going to make mucus, which is a protein, what do you need? You need a UPS or FedEx to ship the mucus, right? Golgi bodies. If you're making a lot of product, you need rough ER and you need Golgi apparatus. 38. Oh, you guys are gonna be bad. 15 of you got this wrong, so half. Pseudostratified cuboidal epithelium is not an epithelial classification, now is it? How many guys think that's a trick question? Go ahead, put your hands up. It's okay. Yup. Read the question. How many guys read that and went, that's no such thing? Good job. The other guys, read question. I totally get why you did it though. You're trying to go fast, right? These tests are balanced and try to go fast and not go too fast. 39, single cell layer of epithelium that forms lining of serous membrane is, yeah, it's squamous. That's kind of a memory thing. 40, 10 of you got it wrong. Best describes connective tissue. Well, doesn't line body cavities, does it? Not a single layer, is it? So, large amount of matrix or secrete stuff? Large amount of matrix. We talked all about the matrix, right? 41. 5 of you missed 41. Matrix is fibers and ground substance. Remember, I had you guys write out, in your notes, extracellular matrix equals ground substance plus fibers. If you got that wrong, either you made a mistake or you're flat not studying your notes enough. Only way around that one. I don't mean to be mean to you. Hey, wait till we get to lab. There's probably gonna be a stat speech in lab. 42. Fiber that gives connective tissue its strength. What fiber is strong? Collagen. Most of you guys got that
I
right.
had it.
43. 11 of you missed 43. Glands such as the thyroid secrete their product directly in the blood rather than ducts are called endocrine. By definition, ductless glands secrete their product directly into blood. Yeah, an endocrine gland, for example, your pituitary gland, or any endocrine gland, the cells just produce the products, and it diffuses into the bloodstream. The pituitary gland actually has blood vessels leading down to it and leading away from it. So that's how those move. I do the opposite. I remember exocrine, exocrine and endocrine are two different kinds of glands. I remember exocrine, exo's outside. So to secrete outside, you gotta have a tube. And that's the duct to get it outside your body. And then endocrine's the opposite. Yeah. How many guys tried to remember hypertonic, hypotonic, and exotonic? Each one remember it. What I do is I just remember one. I have a crappy memory. I can't remember too many things. So I just remember one thing and I rebuild the wheel from that one thing every time I need it. I remember hypotonic is like a hypodermic needle going into the cell, blowing it up. I have that picture in my head. Hypotonic, hypodermic. Two words go together, hypo. Poof. I say, okay, in a hypotonic situation, water moves into the cell and blows it up. Hyper is the opposite. Water moves out of the cell. What would suck water out of the cell? More dissolved stuff. Iso is the same. I remember a lot of things because I remember one thing. Think out the rest. Reinvent the wheel every time I need it. That's easier for me to try to remember both. Because if I try to remember both, it's easy to switch them. It's really easy to say, oops, just remember one thing and rethink it out. I am more likely to be accurate. You guys can try that too. If that works for you, cool. If it doesn't, cool. But that's a technique that I use. Alright, so we're on 44. 5 of you missed 44. Blank epithelium appears to have 2 or 3 layers, but the cells are in contact with the basement membrane. Pseudostratified columnar. Wouldn't it have been nice to think about that back in pseudostratified cuboidals? Look for information on the test that'll help you with other answers. 45. Looks like 19 of you missed 45. Which of these is not considered to be a connective tissue? So more than half of you missed it. How many of you missed it because you forgot the knot or you didn't see the knot? Okay, you just boom, there. I mean, there's a lot of connective tissues there, so picking the answer was easy. Now, how many guys pick blood because blood's juicy? Yeah, it is connective tissue by definition. It's its own category. So one's not considered a connective tissue is muscle. How many guys thought muscle connects stuff together so it can't be that? Come on. A couple of you did. That's all right. 46. First step in tissue repair. Now remember, we talked about inflammation was not the start of tissue repair. 18 of you missed 46. First step is formation of granulation tissue. It's capillaries there. Question?
On 45, was the correct answer adipose?
No, adipose is a connective tissue. Correct answer is muscle, which is not considered a connective tissue. The muscle is definitely not a connective tissue. Muscle is its own thing, its own tissue. Okay, so we've got 46. 47, you guys had a little problem too. Select the correct statement regarding tissue repair. During tissue repair, let's see, yeah, it's another process of elimination. Granulation tissue is not a blood clot. Granulation tissue is a finished product. What is granulation tissue?
Explain.
Brand new, bumpy, pink, fresh skin full of capillaries, right? Very tender. If you guys have split open a cut that you thought was healing already because you did something stupid. Yeah, granulation tissue is not tough. And let's see, clot is not formed by lymph fluid and transposed reticular fibers. That's garbage. So inflammation causes capillaries to dilate and become permeable. And then 48, see 13 of you missed 48. The correct statement regarding epithelial tissues. Again, we can process and eliminate this. Pseudostratified is at least 2 layers. Is that true? No. Pseudo, fake stratified, 1 layer. Simple epithelial form impermeable barriers. No, they're easily permeable. Stuff moves through those guys all the time. Stratified epithelia are tall and narrow. No, that's columnar. Stratified epithelia are present where abrasion, protection from abrasion is important. Your skin, stratified squamous epithelia. You need protection from abrasion. There it is. Alright, now 49 and 50. Looks like 15 of you missed 49. A lipid, that's that thing with 3 wigglies on it. 3 chains. Then 50, polysaccharide. That's the the long chain of circles. Poly, many. E is protein, A is glucose, and E is nucleic acid. Questions, comments, concerns?
They're not the second guess.
Yeah.
Thank you,
Did this
thank
help
you.
you guys at all?
Yes.
Yeah.
Okay, now, I don't curve tests. Because if you guys get an 85 average on a test, that means 85 is a C. You want that to happen? If the test has an 85 average, that means 85 is a C if I curve.
No,
You want that to happen?
no,
Say no.
no.
Okay, I don't curve tests. I do, however, give competition adjustments.
I love it.
I understand that you guys are in competitive programs with other people. Grades matter. And I happen to think that my B students can kick anybody else's A students' butt.
Yeah.
I'm like that. So what you guys thought there were 3 questions. And you guys got confused.
I just misread.
The rest of the tests are gonna be like that. But for this test, take your number correct, put it over 46. And if you got more than 46, you're stuck with your 100, I feel bad.
Oh.
All right? Now get your phones out, do the division. That's your grade. The next question is gonna be, do I round up? I don't round anything. These numbers go in the grade book at the end of semester. So if you got an 89.5, you tell everybody you got an A, cool. Got an 88, tell them you got an A too. At the end of semester, I'll go through your grades and look at them. I reserve the right to raise your grade if it's within half a percentage point. 89.5 and above, I reserve the right to raise your grade. But it doesn't mean that I do it all the time. You consistently annoy me. If you never show up to class, yeah, you get a B. You show up to class, you don't piss me off, you'll get the A. Alright? None of you guys have made me upset. You're all good, okay? You're all good. Fair enough?
Yep.
Time is, we got a couple minutes. Any questions about this? Anything you wanna talk about?
Is there like the organization of the healing process? And my notes I wrote down as you said it, but I had it in a different order. That's why I got it wrong on that.
What was your other
I
answer?
fiberglass multiply rapidly and then form aggressive granulation. I was doing.
Fiberglass are going on at the same time.
Right, and that's what I thought, but. I just wanted to understand more about tissue repair.
You're talking about first step, right?
Yeah. So granulation tissue formation would be the first step.
Yeah,
For fiberglass.
yeah, go ahead and open up your notes here.
Yes, sir.
I mean, your books on that page. I'll let you do it, 'cause I don't know where your page is.
This is a paper that you guys, this is dumb, I wish I had a book, I really do it. $200.
Alright. It's like 2 pages away, there we go.